Hmm. So, good day, students, and welcome to our class on ZEB 462. Okay, animal molecular biology and bioinformatics. So, for the purpose of this course, I'll be teaching the bioinformatics. So, before I start off with this, I'm sure most of you are conversant with the recent developments whereby where a popular musician just passed on named Mubad, and a lot of controversy surrounding his death. Okay, and recently some people are already calling for the DNA test of Mubad's son, alongside with the deceased Mubad, to be sure that the man is truly the father of the son, because there are a lot of suspicions, surrounding the way the wife actually treated him before he passed on and all of that. So now, before I dive into it, bioinformatics is just where you have a system known as algorithm. Okay, you have output, you have input. This input can be known as query, sequence. This is reference, sequence. Don't write, just look and just observe. Okay, query, sequence. So what am I trying to say? Just like having your calculator, you press 1 + 1. This is the central algorithm that will produce 2. Okay, and for the purpose of this course, when I'm talking about sequence or bioinformatics, we are talking about amino acids and what? Nucleotides. Okay, nucleotides, we're talking about DNA or RNA. Okay, so now what you feed into the algorithm to have an output. So now looking at Mubad's case, so how do we link it to bioinformatics? So usually a DNA sample or a sample will be collected from him. To get the DNA sample, one could get sample from blood sample, maybe through the hair or through the saliva, or if he had an already stored sample anywhere. Okay, now those things will be taken to the lab. The DNA will be removed from other cellular components. You still remember when you have a human being, which is the system, you still have organs. You have, from organs, you have tissues, then you have cells, group of specialized cells, tissues, organ, then combination of the other organs make the system. So when you have the cell, you have other component. You have the component, the animal cell, for example. You have the cell membrane and the organelles. So other cellular components will be removed to get the DNA. Now, when the DNA is gotten, it's usually amplified using polymerase chain reaction to get a segment of that particular DNA. So the same thing will be done on the child. Maybe collecting a sample from the child's saliva as well, going through the same process. So matching it, the child's own could be the reference one, while Mubad will be there. So when you now place it side by side, you'll be able to read whether they are actually linked together, whether the child it is or not. So that's exactly what we'll be looking at as regard to bioinformatics, whereby you perform some experiments in the lab, you isolate some DNA particles or amino acids, you put it in a database that has a certain algorithm, and it will produce, search through to produce, a suitable match, a suitable match that will tell you a lot of things about that, so that you'll be able to predict, okay, maybe this particular gene has something with the reference, whatever that came out, that may have his own particulars. You're able to say, okay, this could also be, maybe this particular, the experimental one, which is the query sequence, there's a genetic trait of, say, albinism, or a genetic trait of anything at all. Okay, so that is what bioinformatics is all about. Okay, so now, having had this slight introduction or overview, so we're looking at learning objectives, and at the end of the class we'll be able to look at brief history. We'll look at definitions. We'll also look at data types and databases. We also look at sequence alignment. And importance of bioinformatics. Okay, so by the end of the class we'll be able to achieve the following: a brief history, some definitions, data types and databases, perform some sequence alignment, and also look at the importance of bioinformatics. So we'll take the first one. Brief history. So the history of the bioinformatics dates back as 1962. So in 1962, you have Zukando and Pauling. Both of them, they proposed the theory of molecular evolution. So they proposed the theory of molecular evolution, saying that you have similarities exist between proteins that have the same function. So we started with these two scientists, proposed the theory of molecular evolution, saying that there exists a similarity between proteins, and as at that time, proteins were well known. The issue of proteins and amino acids, nucleotides came, that should be in the late 90s. Okay, so as at that time, that the structural component of, say, an animal cell, where you have proteins performing different functions as regard to, providing structural support, like the cytoskeletons, okay, and other like transcription, translation, and the rest. So at that time they proposed this, and this is what you have. Then by 1965, the likes of Margaret O. Dayoff and her colleagues. Now, Margaret Dayoff and her colleagues, because you have, as at that time, because you have an explosion of experimental data on proteins, you know, somebody will perform an experiment, maybe write it somewhere, put it somewhere, and also there was no, we never had the management of these proteins scattered everywhere. So she and her colleagues had to catalog protein sequence and structure in Atlas of Protein Sequence and Structure. Okay, so that with these people, then Margaret and her colleagues, putting it in a kind of big exercise book, a catalog, so whereby when you open the book, you see different sections: protein sequence, protein structure. So as at that time, we never had a computer, a database where all these sequences are housed. Okay, so it was just scattered everywhere. Maybe I perform one experiment in and I have my data there. Somebody will perform an experiment in another school and all of that. So you can just go here and then get see data littered everywhere, biological data littered everywhere, protein sequence and its structure. So but these guys came and we are able to collate any possible protein sequence and structure at that time and put it in a catalog. Okay, so and having this gave rise to what we have as the computational biology. So it was the advent of when we now started having computers where these sequences are now housed in a database and all of that. So then 1965, and the next one was in 1980. Okay, the EMBL, European Molecular Biological Laboratory. So in 1980, the EMBL established a data library to organize, collect, and interpret nucleotides. So as you can see the protein sequence came before the nucleotides. So the European Molecular Biological Laboratory, based in Germany, okay, they established a data library where one can actually collect and organize, collect, and interpret info on nucleotides. Okay, on nucleotides. So by 19 84 or thereabout. Then you have the NBRF, the National Biomedical Research Foundation. Okay, so these guys, what they established the PIR. The PIR known as, sorry, I need to use this, Protein Information Research. Protein Information Research that allows scientists to identify and interpret information on proteins. Okay, see the movement from EMBL to National Biomedical Research Foundation. Okay, then by 1988, we have the NBCI. That should be the NCBI, not NCBI, the National Center for Biological Information. So they established the Gem Bank. So the Gem Bank was used to collect all the possible nucleotides and proteins available as at that time. Okay, then another group about that same time, you have the DDBJ, the data DNA Data Bank of Japan. So this guy is also they also followed in collecting biodata. Okay, now having all of this, by the same year, the EMBL, the Gen Bank, and the DDBJ, they all came together and decided to be using a single format known as the INSDC. The International Nucleotide Sequence Data Collaboration. Okay, so this is, you have the Gem Bank here, you have, say, the EMBL, you have DDBJ. So all of them are sharing information using this as a central server. Okay, I said the Gem Bank, USA, Germany, and Japan. Okay, so that brings us to the end of the brief history as regard to the nucleotides, the advent of the nucleotides and nucleotide sequence and the protein sequence. So having known all of this, we'll now define proper. Okay, so definition of bioinformatics. So from what we've discussed so far, bioinformatics can be defined, it is a multidisciplinary field that uses biology, computer science, and statistics to manage and analyze biological data. Okay, so multidisciplinary words field, and like I did say, if you remember, where you have this, now you have this is a combination of the computer and you have statistics inside, and you feed in with the biological data. This biological data can come in form of amino acid sequence or nucleotide sequence. Or protein structure, or even what do you call it, gene expression. But for the purpose of this topic, we are just dealing with amino acid sequence and nucleotide. So when you feed it into the computer, will interpret it depending on the type of algorithm, will do the statistical analysis and produces an output. Okay, so that is all for the definition. I hope you are following. Okay, if you have any questions, you ask me on our WhatsApp group. Okay, now having defined this, the next one is we look at so you see biological data. Okay, biological data. So let's look at data types. Data types, you have primary, you have secondary, you have tertiary. Under primary data, like I said, we have the nucleotide sequence, which is DNA or RNA, or the amino acids. You know, the amino acids, valine, leucine, isoleucine, the rest of them. Then for secondary data, you have motifs of protein. Then for tertiary, you have protein structure. Okay, so these are the types of data under bioinformatics. Okay, with reference to just amino acid and what proteins. So now this data, they are housed in what? In a database. So a database is always known as an electronic filing cabinet, whereby you store a large amount of data having this. So let's look at the database. The database. So our database, you have. Let's look at the types of database we have. Like I did say, we have the nucleotide database, and you have the protein or the amino acid. Amino acid. Okay. Now under the nucleotide database, under the amino acid, you have the primary. I also have the secondary. So for nucleotide, we have the EMBL, you have DDBG, the data bank of Japan, European Molecular Biological Laboratory. Then you have the Gen Bank. This is USA, from USA, Japan, Germany. Do we have one in Nigeria? I don't think so. Okay. Now for the amino acid, the primary ones, we have the PIR, protein information resource. You have the Swiss prots, and you have PIR, Swiss prots, and you have unit prots. Then for the secondary, you have CATH, you have PDB, and you have the scope. The CATH is known at classification, full meaning classification, architecture, topology, homology. Then PDB, protein data bank. Then SCOP is a structural structural classification of proteins. That's true. Structural classification of protein. Okay, so you see, let me do a recap of the things we've done. Talked about, started off with a brief history, how we have an explosion of proteins, protein sequences, amino acid sequences, littered everywhere. Or rather, how the works of Zucandol and Zucandol and Pauling actually, gave rise to the theory of molecular evolution, giving rise to how Margaret and her colleagues, tried to catalog, the known protein sequences at that time, then which gave rise to computational biology, whereby these ones came on board, the MBL, DDB, and GenBank. I remember these three are housed in INS DC. Yeah, I think the EMBL, they've added something, I think EBI also, to their own acronym. So if you search this online, just press EMBL website. If you have any type of DNA sequence, if you just input it, it will just run it through and give you possible matches. You can try it, so you can just put. Remember, you're not talking about nucleotides, DNA, RNA. You're talking about the G plus C contents and the A plus C contents, guanine, cytosine, adenine, and thymine. So if I have like A, T, C, G, something like this, I could just use it as my own query sequence, put it inside the MBL website. They will run a search and see if there's something that matches, then they will tell you, if there is a possible match, you see, the genetic, specific genetic information that the reference sequence will have as regard to this particular one. So that's all about this. Then the next one is what again? You have I didn't even explain. Okay, you have motifs. Motifs are motifs of protein. You can also have motifs of what do you call it? Nucleotide sequence. Okay, so motifs are, if you have, say, the amino acids, maybe valine, leucine, isoleucine, have a stretch of the amino acid sequence. Let's just say it A, G, U. If you have something like that, as I don't have all the amino acid sequence of that, maybe have a stretch of it. Maybe in between this, you have simple structures of protein. Now, this could be a conserved sequence. Now, this conserved sequence may be telling us something that is very unique about this particular stretch of sequence. Maybe the albinism or Down syndrome, anything at all. Okay, so it's just short sequences. Then obviously the protein structure has a 3D dimensional shape. Okay, with this we'll now go to sequence alignment. Now sequence. Now remember, I did say that when you have this as the algorithm, you have input, you have output. Okay, so we'll look at what actually plays out inside this particular algorithm. We have different algorithms, that tend to analyze sequences to produce an output. So under alignment, sequence alignment simply means defined as a bioinformatics technique used to compare and analyze two or more sequences in order to get the best possible match. The technique used to compare and analyze two or more sequences in order to get the best possible match. So that is sequence alignment. Now, under sequence alignment, we have two types. You have the pairwise and you have the multi-sequence alignment. but our focus will be on the pairwise. On the pairwise, you have the global alignment, and you have local alignment. Global alignment and local alignment. Now, the global alignment uses the algorithm. It uses is needle man wunsch algorithm. Okay. Well, I just want to use it Smith Waterman algorithm. This one is for global alignment, is usually for sequences of equal length, while this is used for short sequences. Okay, sequences of equal length. So I'll explain how some of these stuff work. Okay. So okay. Now for global alignment, let's have a query sequence from the lab. That's the experimental sequence. Okay, we want to analyze say A, T, G, G, C, T, A, C. Okay, then the reference sequence should also be 1, 2, 3, 4, 5, 6, 7, 8. We may have C, T, G, C, T, A, G. So in global alignment, you will make use of, like I said, remember the algorithm. The algorithm is needleman wunsch. That is what is here. That states that for you to perform global alignment using this particular algorithm, that the sequences, these are the nucleotides. Remember your base pairs, adenine, thymine, guanine, cytosine. They should be of the same base pairs, whereby the query sequence is the one you attribute. This is the reference sequence. So by the time you check and you see if you get a possible match, well, usually this is just an example. I'll use a local alignment to explain further. Okay, you see A to C, no alignment. T to T, there's an alignment. G to G, alignment. G to C, alignment. T to C, this one, no alignment. Okay, no alignment. Hold on. Okay, no alignment, no alignment, no alignment. Okay, they are not complete. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6, 7. 6, 7. Okay, maybe C. Then alignment. Okay, so maybe within these two places where there are no alignments, global alignment tends to give us so that you have a global picture of what to expect. Where if there is conserved, just like you have conserved, if there are motifs, conserved sequences, that'll tell you, something about that particular, the two sequences that are being aligned. Okay, then for local alignment, that will be our focus. for today, so use it to explain better. So usually, okay, don't worry. So for local alignment makes use of, okay, if this is your reference sequence, say ATGCTAT, then you have your query sequence. It could be, let's say, let's so that I don't make it too tall. We have 1, 2, 3, 4, 5. We have A, T, G, C, T. Okay, 1, 2, 3, 4, 5. Call this 1, 1, 2, 3, 4. You have this as 2. So local alignment, instead of having sequences of equal length, you may have this stretching to a certain length. It's just for you. You can use, and maybe you have a short, what do you call it? A short sequence. You can use the short sequence to place, look for the best possible match, and that is the essence of sequence alignment, to get the best possible match. So that is why local alignment is more advantageous to global alignment. For under global alignment, the two sequences must be of the same length. Okay. Now for this, one will say you can solve this particular problem, generating the best possible match. Now in local alignment, you make use of binary coding. Okay, we'll say for match, we'll have +1, mismatch, we have -1, then for gap, we'll have 0. Okay, so let's solve. You have A, T, T, G, G, C, T, T. So you see, there's a match here. There's a match here. There's a match here. There is a mismatch here. Then there is a gap. So 0. So match, you have +1. Okay. Instead of having because they will cross. So let's just use 2. If you use 1 - 1 will give you 0. So let's just use 2. So for match, we have +2. T plus T, you have 2. G and G is a match, you have 2. C and T is a mismatch, you have -1. Then if there's a gap, you have 0. So if I add all of them together, what do I get? 2 + 2, 4. 4 + 2 is 6. 6 - 1 is 5. We have 5. Okay. Now we want to see if we get another type of match. The same ATGCT. We can push this to the other end, whereby you have ATGT. Okay. So in solving this, you have A to this, there's a gap. So for gap, you have 0. This is a mismatch. Mismatch is -1. This is a mismatch, -1. This is a mismatch, -1. This is a match +2. So when you add -1 - 1 is -2. -2 - 1 is -3. -3 +2 is what? -1. So the same way, just keep interchanging them until you get the best possible match, which is for now we have 5. So I don't know if there's any other configuration. It's just what you test. So this is the practical way what happens inside that algorithm. So when you carry this and place here, the algorithm will run through and try to provide or produce the best possible match instead of going through this tedious procedure. Okay, and some of you, -1 to still remember directed number. Let me teach you basic mathematics just in case you may have forgotten. Just be wondering how we got -3. Where you have your directed number, this is 0. You have positive infinity, negative infinity, +1, +2, +3. This is -1, -2, -3. Okay, so for example, if I say 1 + 1, the answer is 2. How did I get 2? So the first one is your start button. The sign will tell you left or right. This is also you're going right. Then the other sign, the other number will tell you how many times you go. Number of times. Okay. So we'll start from 1. We'll go right plus another 1. That is 2. So that's how 2. So if you have -1, -1, the first sign is -1. Start here. We go left. How many times? Once we have -2. Okay. Directed numbers. That is in New General Mathematics. I think in JS1 or so, chapter 17. Back then. Okay. So this is alignment of sequence alignment, global and local alignment. So the essence, like I said, to provide the best possible match, and using the computer, using statistics, to give us the best possible match. So the last part is the importance of why do we bioinformatics. So from what we've done so far, is number 1, to manage vast so a vast explosion of biodata. Okay, understanding complex systems that occur in the cell. I'll explain. To understand evolutionary traits. Okay. Use in drug production. And so many more. Okay. So like we did say, the bioinformatics, like I said, everything is stored in the database we use to manage and store a lot of biodata or biomolecules, like the gene, DNA, the RNA, gene expression, and the likes of it, even robotics. Okay, then use in understanding complex systems that occur in the cell, like such as you have metabolism. I think we did this in our second year. You have your transcription, okay, and you also have translation. I'm sure you've done this in your genetics. Transcription, where you have a DNA strand, you know, if you say A, T, G, C, you know, then you have your messenger RNA. It's a transcription translation are involved in protein synthesis. Okay, this happens inside the nucleus, while translation is in the cytoplasm. Okay, you know, A pairs with what? For messenger RNA, A pairs with T, but since we don't have T in messenger RNA, or we have this, then T pairs with A, G pairs with C, C pairs with G. So it copies a complementary strand of this. The messenger RNA will move out of the nucleus to the cytoplasm where you have the ribosomes and translate it to protein. I'm sure you guys did this in your third year. Okay, understand evolutionary traits, genetic traits. Okay, just like I said, why a certain family tree or a certain family, why are they intelligent? You know, there could be a reason for that. Everybody there is intelligent, or maybe albinism moving, or why, you know, this family, they have twins in their bloodline, or anything at all. Then using drug production. Okay, during the COVID period, you know, people went into the lab trying to, you know, produce different sequences and looking for possible match to use to develop drugs and all of that. So with this, we'll come to the end of the class. So a recap of what we've done so far. We looked at bioinformatics, you know, looked at the brief history. We defined bioinformatics, the data types, the database, sequence alignment, the different types of sequence alignment. We have the global alignment and the local alignment, you know, conditions of using global and local alignment, and the importance of bioinformatics. Okay, so come to the end of the class. Have a good day.